Good morning. Good morning, Caltech alumni, family, and friends. Come and sit down and join us. Yeah, we're back. Alumni Day, it's back. We're here after the pandemic. It's been since 2019, so welcome. First, I want to say it's happening live in Pasadena right now. And I'm going to ask you as you come in to please silence your cell phones, but do leave them on, and we'll tell you why in a moment, and just note your emergency exits. But I should say, it's not just the 300 of us here in Beckman today. There are over 400 people joining us from techers from all around the globe, and they can also check their emergency exits if they wish. I am Sandra Tsing Loh, Caltech alumna, BS Physics 1983. Any 1983s here? Okay, all right. And host of the Daily Radio Minute, The Lowdown on Science, syndicated by NPR and KPCC, now called LAist, a show actually created by Caltech almost 20 years ago. By then PR head Bob O'Rourke under the tutelage of then President David Baltimore. Who is brilliant and has a sense of humor too. I'm also a proud member of the Techer community of more than 25,000 alumni living and working all over your planet. So I'm gonna be hosting this first ever hybrid Alumni Day, where we'll be here and people are watching elsewhere, as I said. Speaking of which, our in-person guests today for this exciting, informative, interactive, and fully Caltech event, here to open the day officially, now that you're seated, I bring you the Sonia and William Davidow Presidential Chair, Professor of Physics, Caltech President, Thomas F. Rosenbaum. A warm welcome to you, Tom, if I can call you Tom after that. Okay.
Thank you. Welcome to all. I want to start with a shout out to Sandra. So thank you so much for doing this. We've been playing Hollywood Squares for the last few years, but she's a mainstay of these events now, and it's just great to be able to do it in person. I also want to thank the speakers. Where else but Caltech on one day can you get more than 3% of the faculty engaging with our alums? And I want to thank all of you for showing up, and those online as well. Nearly 1,000 people, or we'll get there, all together, sharing in the intellectual repast that we have to present. So I was coming out of Parson Gates, where my administrative office is, the other day, yesterday, and I saw a number of tours. Some of you may have been part of them. And I was thinking that the campus has really changed. Of course, for those of you who go far enough back, the campus ended at San Pasquale. It's now, of course, extending up to Del Mar, and in fact, you'll find Caltech buildings north of that. But a lot of new buildings, if you haven't had a chance to walk around. The Hemetman Center is a gathering place for all members of the community, has a rehearsal hall in it. It has a much expanded Red Door Cafe where you can just sit at a table and hear all kinds of great conversations going on, all about science and engineering. New undergraduate residence, the Bechtel residence, which allows us yet again now to have all our undergraduates on campus for all 4 years, which I think is a really important part of the experience here. If you go to the northwest corner, there's a new Center for Neuroscience, a big copper building, which has lots of interactive spaces, which brings together members, students, faculty, postdocs, staff from all 6 divisions. And if you go inside now the Lind Hall of Mathematics and Physics, which used to be Sloan, and Gates Thomas, which houses mechanical engineering, you'll see a complete gut renovation where the facade has been preserved. And there's more to come. Come back in a few years and we'll have a new Center for Quantum Precision Measurement. The panel, I think you'll hear about that, how this is an organizing principle, quantum mechanics, to bring together individuals from lots of disciplines, chemistry, biology, computer science, physics, applied physics, engineering, who want to use entanglement to be able to build new devices to better interrogate nature. And we also will launch next year the Resnick Sustainability Institute building, which is on the west side of campus as a new entrance. And importantly, on its second floor will house all new freshman chemistry labs, which now will be sustainability laboratories. So a lot of change. But the essence of Caltech remains the same. And you can look at Gordon Moore as a wonderful example of that, right? Pioneer in the semiconductor industry, thinking of him because, of course, he passed away in March. And he said, if everything you try works, you aren't trying hard enough. And I think that's just perfect. It's how we've been. It's how we challenge ourselves, right? If you get an easy solution, you aren't pushing yourself hard enough. You aren't asking important enough questions. You aren't switching to a new field, which may be more productive than what you're working in now, even though that's a hard thing to do. We see that for our faculty, we see that for our students, we see that for our alumni. And this characteristic of pushing ourselves together to discover something new about the universe, to develop technologies that improve people's lives, continues to be an animating spirit here at Caltech. So thank you for coming today. This is a statement of community. It's the alums who can make it here to Pasadena in person and exchange ideas and reminiscences and notions of what's important in person, and those online who are sharing their experiences through the Q&A and also through future interactions that make this such a powerful place. So thank you for that. Enjoy today. And I'll look forward to seeing you at many seminar days and alumni reunions to come.
Thank you so much, President Rosenbaum. Inspiring words. And so speaking of pushing ourselves as a community today, before we begin our first of 6 fascinating sessions, I'd like to introduce you to our new friend, Slido. Applause for Slido. You don't know what it is, but we're voyaging into the unknown. So today, your Q&A will be facilitated via, yes, your phone. Okay, so let's take a deep breath. If you go to, you see that QR code? I want to say, good, phones are going up, checkers, good, okay. So if you go there, it'll take you to the app Slido, where you can ask questions while our first speaker is speaking. And for instance, and if you don't feel like typing on your phone, you can actually ask. You can actually say, for instance, I would say, quick demo, I've been here at Alumni Day since 1965 as a child, so I would say, where are the donuts? Send, okay. Now, it'll take a moment if you're looking at the app, which I hope that you are, for them to okay the question, and then it will come up, and then there it is, say where are the donuts, and then you can like on the right, you can actually hit a like if you like someone's question, and it'll move up in the queue. Of course, while our first speaker is speaking, do not ask where the donuts are. They're on Saturn, I think you saw out there. So these are actually questions for our speaker. And of course, we want you to be focusing on the lecture, but also if you have a question that goes along the way, you can ask, and we'll be able to address it likely in the Q&A. So, and if you can't log on, you can go to slido.com and enter, and the code is Caltech1. Case doesn't matter for the first seminar lecture, and then Caltech 2 will be the second one, etc. And we'll be here to support you along the way. Okay, enough with the phones. You should have all the programs with those agendas. Our virtual friends, you can use Slido also, and you can find it on the WebEx platform. Now, here's a brief outline of our day. Now that you've had that great breakfast, we get a little insight into whether we might actually think with our gut. We'll take a step into a fascinating future as we learn how Caltech moves us from quantum theory into quantum science. Since we'll already be in the future, one of Caltech's newest faculty will take us into the world of artificial intelligence and how machines learn what we might really be thinking about them. And if you've been following the ChatGPT bot, I think it'll be especially fascinating. Just before lunch, we'll ask you to hang on for just a few minutes. You'll be the first to hear who's actually been selected for the 2023 Distinguished Alumni Awards, and you'll see an actual film premiere. Don't worry, it's just a few minutes long, but it's very exciting. We'll break for lunch. Those of you on campus can get the grab-and-go picnic lunches and explore. Lots of facilities are hosting open houses, which you can find in your programs. Those of you at home, I direct you down the hall to your fridge, to your microwave, whatever. Just be sure to check your emergency exits in your own homes. But please be back at Beckman or at your screen by 2 p.m. after lunch, where we go where no researcher has gone before, exploring extraterrestrial environments on our solar system and beyond. We'll explore our hidden, this is really a favorite of mine, I have to say, unhidden fears with expert from the Fear Lab at Caltech. I don't know if you went to Caltech, if you've gone to Caltech, the whole thing was a Fear Lab, but anyway. Phys 106, anyone? AMA 95, anyone? I'd be interested in hearing what courses were especially fearful for you. For me, it was all of them. Even Computer Science 1 was. Anyway, in case you feel like you're running out of steam at the end of the day, we'll end the day discovering how Caltech is driving new technologies with renewable energy. And welcome. Each talk will be followed by a 15-minute break, so you get a snack, some water, a donut, and please remember to use your new friend Slido so you can pose questions to our speakers today. And the first is Caltech 1, and you can try it out. Okay. So our first session, indeed, is the gut-brain connection with Dr. Sarkis Mazmanian, the Louis B. and Nellie Sue Professor of Microbiology and Heritage Principal Investigator. Dr. Mazmanian was born in Lebanon to 2 Armenian refugees. His family moved to the US in 1973. He enrolled at UCLA in 1990, planning to major in English, before switching gears to graduate in microbiology, earning his PhD in 2002. Affiliated thereafter with the Harvard Medical School and University of Chicago, Mazmanian won a MacArthur Genius Grant for his work on the human microbiome. The founder of 2 biotech companies and so much more, Mazmanian has explored the fascinating relationships between humans, bacteria, and myriad vaccines, antibiotics, and sanitary practices we put in place to control our environment, or at least attempt to. The question, how does gut bacteria influence the development and function of the immune and nervous systems? Today, Dr. Mazmanian leads us on an examination of the gut-brain connection. Welcome, Dr. Sarkis Mazmanian.
Thank you for that amazing introduction and the opportunity to speak here today at Seminar Day about our work. I will tell you about a story that has taken well over a decade to evolve, and you'll appreciate it went from an initial idea to what is now an active clinical trial. And so I'm going to just walk you through the basic science and how that's informed our hopes to improve human health. So the title is The Gut Microbiome and Autism Spectrum Disorder from Discovery to Drug. And so I'm a microbiologist by training. I work specifically on bacteria and even more specifically on gut bacteria, the many trillions of cells that live in and on our bodies, most of which are in our lower gastrointestinal tract. So if we actually look at the numbers of bacteria inside our colon, where again most of them reside, we have about 100 trillion bacterial cells in us. And what that means is that we are mostly bacteria on a cellular level. There are more bacteria in our bodies than there are human cells. By some estimates, about 3 to 5 times as many bacteria. And even more astounding are the number of genes, right? So the microbiome refers to the actual genes that are harbored in these bacteria. There are about 150 times as many gene families or unique types of genes. So we view our microbiome and our genome as a collaboration that come together to affect our health and in some cases impact disease. And I'll tell you about one of those examples. And the microbiome has been referred to an alien or foreign organ because it has the metabolic capacity of the liver and the actual weight of the adult brain. And so in a number of different clinical and preclinical settings, the microbiome has been linked to neurological, metabolic, and immunological conditions. And I'll just list them here, and this is really just a small sample of the types of different conditions and in some cases diseases and disorders where the microbiome has been implicated. Most of this work is still early, meaning that a lot of the work is still in preclinical models, mostly mouse models, but other model systems as well. And we are now, as a community, beginning to translate some of this work and apply it to clinical settings. But I have to say there's a lot of excitement because the data, at least in the animal models, shows that the microbiome is a critical contributor, in some cases causal, to the development of disease, and perhaps we can get similar data in humans and give us some actionable items to improve human health. And so the story I'll tell you about today, it revolves around autism spectrum disorder and some of the behavioral symptoms that are associated with autism. So our laboratory works on a number of different conditions, again in animal models that span Parkinson's, Alzheimer's disease, and anxiety. We started working on autism very early on, actually just a few years after I joined Caltech, and I'll tell you that story in a minute. And so autism spectrum disorder is defined by core behavioral traits that include deficits or reductions in social communication. That includes verbal and nonverbal forms of communication, as well as repetitive behavior. And that's what's shown here in these core symptoms. But there are many comorbidities or associated behaviors and phenotypes associated with autism, and those are listed here on the periphery. And what caught our eye many years ago were these gastrointestinal symptoms. And again, about 13, 14 years ago, there were just some initial reports, largely anecdotal, suggesting that there may be an increase in GI symptoms such as constipation, diarrhea, bloating in the autism population compared to that of the healthy control population, as well as immune dysfunction and metabolic abnormalities, which are associated, as I just mentioned, with the microbiome. So we thought perhaps there was a link between gut bacteria and even the gut and the development, or at least the progression of autism. And of course, there's a number of different associated traits with autism, and those are listed here, and I'll just draw your attention to anxiety and to aggression, because those will become important in a few minutes. So the prevalence of autism is rising. The latest estimates from the CDC indicate that 1 in 36 births in the United States result in an autism diagnosis. And you can see the really steep trajectory of the increase in diagnosis of autism. There's controversy in terms of what is driving this increase. At least what we know is that increases in awareness and changes in diagnostic criteria have led to a large portion of this increase. But there are still aspects of the rate of increase of autism that do not remain explained. And I will suggest that the environment plays some role in that, and, because our environments have been changing over the last several decades, and perhaps that's contributing to autism. But I also want to be cautious in mentioning that this is still a hypothesis. Despite a lot of data from many labs around the world, we still really don't know what are the environmental components or the environmental risks associated with autism. But clearly this is a growing social, medical, and economic burden. The current estimates are that there's up to 75 million people worldwide with an autism diagnosis. And this is an estimate of the actual prevalence of autism, largely in the developed world, because it's quite difficult to diagnose autism. There are no blood tests that are reliable. There are no brain scans that are reliable for diagnosing autism. This requires trained behavioral individuals or people with behavioral training to assess autism based on standard questionnaires and other diagnostic tools. And so these are very hard to implement in developing countries. And so one may wonder that the actual incidence of autism may be greater than what we actually know. And as I've already mentioned, the rate of autism is increasing, and this makes the rapid rate of increase makes autism the fastest growing neurodevelopmental disorder. I've mentioned the economic burden already. In a person's lifespan, about the cost to both society and the individual is about $2.5 million. That's including both therapy and lost wages and opportunities. And again, there are these gastrointestinal issues associated with autism. And we now know that the microbiome, the collection of bacteria, viruses, fungi, and other microorganisms in the intestines of individuals with autism look different than that of people who don't have an autism diagnosis. And so, of course, this is an association, that there's a change. What causes that change, what's driving that change, we don't know. And as I've mentioned, the implications of the change in autism on the symptoms itself is something that we and many other people are studying. And so Sandra mentioned, one of the interesting aspects of Caltech, and Tom mentioned how we get out of our comfort zones and reach for the stars in the research that we did. And I became interested in this in a conversation with Paul Patterson. Some of you may know Paul was a neuroscientist here at Caltech. He sadly passed away in 2014. But Paul and I began a collaboration, as I mentioned, very early on in my career here at Caltech. And this is a neuroscientist. Paul was interested in how environment shapes behavior, and a microbiologist coming together and trying to forge new ideas and new discoveries. So what Paul had done when I first started working with him is develop a mouse model of autism that's now widely validated, used all around the world, based on epidemiologic data that an infection during pregnancy, a very severe infection, oftentimes associated with hospitalization, leads to an increased rate of diagnosis in the child when the individual is born. And so Paul modeled this epidemiologic data by essentially giving flu to pregnant mice. And so that's depicted here. And so when the mice had flu, again, during pregnancy, they resolve the flu, the fever goes away, and then when the offspring, the pups, are born to this pregnant mouse, those animals have features of autism. And those features are changes in social behaviors, changes in communication, increased anxiety, and changes in sensory issues, as well as structural changes in the brain in terms of what types of cells move to different regions of the brain as the brain is developing. So this was very exciting to us to be able to have a model that we can test this hypothesis that perhaps the gut was involved. And again, this was quite the hunch when we started working on this, where a new model of autism is still being developed, was the testing ground to understand, is there an implication for the gut? Is there a role for the microbiome in any aspect of autism in this particular mouse model? And I'll just cut to the chase, is that when we first started working on this, one of my students went to Paul's laboratory. They already had an ongoing experiment, and Paul, being a neuroscientist, told me, of course, we throw away the intestines of these animals. We're interested in the brain. And so my student went, harvested the intestines, and on the first experiment, we found some really fascinating results. The microbiome was different between the mice that had the behavioral issues and the mice that behaved, quote unquote, normal, as well as changes in gut physiology. The way the intestines were performing were different in these two groups of mice. And so we had an idea that perhaps one of the ways to interrogate this initial finding was to intervene at the microbial level. And my laboratory at the time was working on an organism called Bacteroides fragilis, which is found in about half of the human population. So half of us here and online have Bacteroides fragilis in our colon. And we had been studying this organism because of its quite strong pro-inflammatory or anti-inflammatory effects, meaning that it was able to suppress inflammation. And the context in which we were interested in Bacteroides fragilis was to correct issues in models of Crohn's disease, ulcerative colitis, and other inflammatory conditions. And I'm not going to go through the details, but we had worked out many, many aspects of the mechanism of this organism. What is the molecule it's making that mediates these anti-inflammatory effects? What receptors does it bind to? What are the cell types that respond? And how does it ultimately suppress inflammation? And so we applied this organism, Bacteroides fragilis, to the mouse model of autism, to this MIA, maternal immune activation model. And then we behaviorally tested these mice, and to our surprise, adding the probiotic, adding this human commensal organism, this beneficial bacteria to mice, corrected many of their behavioral and GI symptoms. So my time is short, so I'm going to just give you a couple of examples of this prior work and then segue into some of our new discoveries. So we tested anxiety in these animals. So anxiety is tested by an open field test where we place mice in this arena. And mice are timid, shy, and anxious animals. They stay close to the walls. You may have already noticed this in your backyard or other places you've seen a mouse, because they just feel safer closer to the walls. But they venture out into the center to look for food or mates or just curious, and then we measure the time the mice spend in the center. The more time they spend, the more bold they are. The less time they spend, the more anxious they are. So these are the animals in the third bars that were the offspring of the mice that were given the flu, offspring of the mice that were given essentially an infection during pregnancy. And you can see they enter the center less than the control animals in the first bar, and then we measure the time they spend in the center, and they spend less time in the center compared to the control animals. And when we gave them the probiotic Bacteroides fragilis, you can see in the third and the fourth bars that these animals now enter the center more often and spend more time in the center, again, compared to their wild-type counterparts, suggesting that we've overcome their anxiety phenotype by just giving this one organism. It's the same with vocalization. Some mice vocalize through ultrasonic communications, and we can measure this using an ultrasonic microphone. And then essentially, you even see patterns of language in these animals. When we looked at the MIA mice, the offspring of the mice that were given flu, you can see that these mice communicate less. The number of calls are decreased, and the duration of their calls are decreased compared to these control animals. Again, a trait that's corrected by Bacteroides fragilis, this one organism. So there were many other aspects of the physiology of the MIA animals that we interrogated and understood in this initial study, but we became interested in what is linking the gut and the brain. How is this signal sent by bacteria from the intestines, presumably to the brain, to mediate these beneficial effects? And so we looked at the immune system, and we found some interesting pathways that were activated by the organism that suppressed inflammation. But then we became interested in the metabolic conditions that are associated with this disorder, because as I mentioned, there's metabolic changes in individuals with autism, which we also observed in the animal model. And so we did an experiment called untargeted metabolomics, where we just surveyed all the small molecules in the circulation of the different groups of mice and tried to understand what is the chemical fingerprint, what's the signature of the different molecules in the blood of these animals that may differentiate how they're behaving and how they're responding to the probiotic. And so these are the control animals. So these are mice just given saline or salt water, not given the flu. And then these are the animals that were given, whose parents were given, or the mother was given the flu. And you can see there's an increase in this one molecule called 4-ethylphenyl sulfate, or 4-EPS. And so here's the molecule here. And this was quite astounding to us. So we looked at a number of different molecules, many thousands of which, found several dozen that were changed. And this was the molecule that was the most highly increased between the control animals and the animals with the autism-like features. And this metabolite was also decreased in those animals that were given Bacteroides fragilis. And what's also really interesting about this molecule, here's the chemical structure of 4-EPS, is that it's chemically very similar to another molecule called paracresol sulfate. And why this was a really exciting discovery for us was that paracresol sulfate had been shown to be increased in the urine of children with autism. So it was a urinary biomarker, if you will, for autism. And again, we had discovered this molecule. There's very, very little known about it, about 4-ethylphenyl sulfate. There were just a couple of papers describing its synthesis, and its chemistry had not been associated with any biological phenotypes, not found in any animals or humans at the time. And so again, we thought maybe this is just more than a coincidence that the molecule that is most highly dysregulated in the animals that have or don't have behavioral issues is very structurally related to animals, or very structurally related to a molecule that's increased in human autism. So this is a mouse discovery, and so the next thing, or one of the next things that we did was to try and validate whether or not this molecule is actually increased in individuals with autism in a human population. Again, the metabolite 4-EPS had not been highly studied at all. And so we were very fortunate to work with Paul Ashwood at UC Davis and Alessio Fasano at Massachusetts General Hospital to obtain both fecal samples and what I'm showing you here, plasma samples from a large cohort of individuals with autism or typically developing controls, and found that about a third of the autism population in this cohort expressed very, very high levels of 4-ethylphenyl sulfate. The molecule is detectable in every single sample, and we've done many studies in humans now subsequent to this, and this has been validated in a number of cohorts now. We all have this molecule, but it's just elevated in individuals with autism. And if we look at those individuals with gastrointestinal symptoms, there's about a 9-fold increase over people, over the typically developing population. And if we look at the autism population that don't have gastrointestinal symptoms, it's a little less than 5-fold increase. And so there's a correlation between GI symptoms, gastrointestinal symptoms, and this molecule. So everything I've shown you to date is, or so far in the talk, is an association. So we identified the molecule in mice. It goes up and down based on a probiotic treatment. It's elevated in populations of people with an autism diagnosis. So maybe it may mark individuals with autism, help us identify who they are, but we will also want to understand, is there a functional role for this molecule? Is it actually doing something relevant to autism, again, whether it's behavioral, gastrointestinal symptoms, or otherwise? And so the molecule, it was not known how this molecule is actually produced, But based on some additional experiments that we did, we were convinced that the only way this metabolite 4-ethylphenyl sulfate was synthesized was through bacteria. We don't have the genes, mice don't have the genes, flies, worms don't have the genes to make 4-ethylphenyl sulfate. And so we worked with Michael Fischbach at Stanford to identify the bacterial genes that can turn tyrosine, an amino acid that is found in our diet. So all of us, as we eat food, ingest tyrosine. And through 3 chemical conversions, tyrosine is metabolized to 4-ethylphenyl. And so this is the bacterial reaction. Again, 3 bacterial enzymes convert tyrosine to 4-ethylphenyl. And then when 4-ethylphenyl is absorbed in the body, it is very rapidly sulfated in the liver to 4-ethylphenyl sulfate. So 4-EPS is the bioactive molecule. And then we perform this chemical reaction because many of these phenolic molecules, these molecules with a ring-like structure, can be quite toxic. And so we sulfate them so that we can excrete them in our urine. And so, again, Michael and his team were able to identify the enzymes, bacterial enzymes that can produce 4-ethylphenyl. And then we engineered groups of bacteria that either produce 4-ethylphenyl, and that's a 4-EP positive group, and another set of bacteria that were bioengineered not to produce 4-ethylphenyl. The same exact organisms, identical in every way, except the 4-EP positive bacteria contain these 3 genes, and then the 4-EP negative group bacteria do not contain the genes. And then we colonized mice with these 2 groups of bacteria. So we made now 2 different lines of mice with that, in theory, the only difference in the entire body, entire chemical signature of these animals was this one molecule. So of course it's an animal study, it's reductionist and it's simplified in many ways, but it's also a very clean system. It lets us get to the biology of this one molecule. And so we showed that 4-ethylphenyl is produced in the feces of the mice that were colonized with the 4-EP positive bacteria. 4-EPS, again, once it's sulfate in the liver, is found in the urine and in the blood of only the group that is producing the metabolite in the intestines. And then very interestingly, we showed that a small population of this molecule, 4-EPS, actually winds up in the brains of the animals. And we think something similar is happening in people. We haven't proven that yet, but at least in these animal studies, this metabolite is getting into the brains of mice. And then we, of course, we were just looking for a phenotype. We were trying to understand what this molecule does. We had really no reason to believe that it affected any particular brain region or any different cell type because, again, it's a novel molecule. So we performed an experiment where we essentially looked at which regions of the brain were more or less active between these 2 groups of mice, relating those activity results to this one molecule. And so in collaboration with Daniel Holschneider at USC, we identified that there are several different brain regions that are more active in the 4-EP positive group compared to the 4-EP negative group. And the regions that are more active are listed here, and those are in red. And there are several regions of the brain that are less active in the 4-EP positive group. And when we saw patterns like this, we wondered whether or not there were changes in connectivity, changes in how regions of the brain were actually communicating with each other. And in fact, we worked with Mikhail Shapiro here at Caltech to do a functional ultrasound experiment to actually show that indeed there were changes in connectivity between different regions of the brain. And why this was so important is that these changes in connectivity have been associated with the autistic brain. And so again, we're modeling certain features of brain activity in the mouse that's associated with autism and other neurodevelopmental disorders. And then in a series of experiments, again, which I don't have time to tell you, we narrowed down the activity of 4-EPS to a particular cell type called oligodendrocytes. So oligodendrocytes are cells that we and mice have in our brains, and these are cells that myelinate neurons. And so neurons or nerve cells have long axons. That's how they send signals over long distances. And what myelin is think of the coating around a copper wire. Myelin coats the long axons of neurons and allows those electrical signals to be sent over long distances. And what we showed is that the maturation state of oligodendrocytes was dependent on this microbial molecule. So as 4-EPS entered the brains of the mice, it arrested or delayed the activation of oligodendrocytes. And then you can see here, the 4-EP positive group have less mature oligodendrocytes than the 4-EP negative group, and we did this by staining and several other methods as well. This is quite profound, that the actual function of this brain cell was dependent on exposure to this microbial molecule. And then we also showed that the reduction in oligodendrocyte activation led to changes in myelination patterns in the brains of these animals. And again, myelination changes are also associated with human autism. So we felt like we were on the right track here. But of course, our main objective was to understand, is this one molecule capable of inducing behavioral symptoms through alterations in myelination? And so we performed a battery of behavioral tests, and showing a test just for anxiety. So this is the open field test that I've already introduced you to. And you can see the mice that were associated or exposed to 4-EP in their intestines, that have 4-EPS in their brains, are less bold. They're much more anxious because they don't enter the center as often as the control 4-EP negative group. And there's no changes in distance moved, so this is an important control experiment, because if the animals were sick, if this molecule was actually making them ill, then they would be less likely to enter the center. But there's no changes in actual locomotion. We interpret this as the mice are choosing not to enter the center of the arena if they're exposed to 4-EP. And then we supported this with additional behavioral tests, and I'm showing the elevated plus maze, where we place mice on a platform that's elevated over the ground by several feet. And so mice feel much safer in the closed arms of the elevated plus maze, but they do go exploring into the open arms, and so we can measure the amount of time that the mice spend in the open arms and the terminus of the open arms, and you can see that the exploratory behavior or the boldness of the animals is decreased in the 4-EP positive group compared to the 4-EP negative group. And so we supported this with a number of different experiments and experimental paradigms to really convince ourselves that indeed 4-EPS was inducing changes in behavior in these mice. And so just to summarize what I've told you is that there are changes in the gut microbiome of individuals with autism that we can recapitulate in an animal model. And there's several other animal models that show the same phenotype. And when the microbiome changes, the molecules that they're producing change as well. So their metabolome, that chemical repertoire of bacterial molecules, is different when a mouse has features of autism and a person is diagnosed with autism. And that leads to changes in the flux of molecules across the intestinal epithelium into the circulation. In the case of 4-ethylphenyl, it's sulfated. In the liver, concentrations of 4-ethylphenyl sulfate build up in the circulation and actually into the brain. And what this molecule does is it arrests oligodendrocyte maturation of those cells that I told you about, leads to reduced myelination in several regions of the brain and altered behaviors. And we think 4-EPS is just the tip of the iceberg. There are many, many molecules that have been identified in humans that are different between a person with a diagnosis and without a diagnosis of autism. And many of those molecules share chemical and physical structural and structural similarities to 4-ethylphenyl sulfate. And so again, we've intensively studied this one molecule, but there are many, many other molecules that we feel either will work with 4-ethylphenyl sulfate or may have different and various different phenotypes that they may induce on their own. And so I'd like to summarize just this basic research that we've done, first using a bacteria and then learning from that bacteria to identify a particular molecule that's mediating these behavioral effects. And so there were changes in the gut. I didn't show you all this data, but in that MIA model that I mentioned, there's this syndrome called leaky gut. So these are the epithelial cells that line our intestines. They keep everything out that may be toxic, but let things in, such as nutrients that we ingest in our diet. And when there's leaky gut, meaning when the seal between these epithelial cells is broken, then more of these molecules can get into the circulation, build up, and get into the brain. Again, this is what we saw in the MIA model. And then when we gave Bacteroides fragilis, it resealed the epithelium and then reduced the number of these the amount of these toxic molecules that get into the circulation. So we're developing Bacteroides fragilis as a probiotic, and that work is well underway. But in the meantime, we thought there was a faster path to perhaps intervening in this chemical communication between the gut and the brain. And so we identified a particular small molecule drug that binds these metabolites, that binds 4-ethylphenyl sulfate. And I'll introduce you to the drug in a second, but our hypothesis was, again, that we've learned about this chemical transfer between the gut and the brain through these basic research studies, through using a probiotic, but now that gave us the opportunity to intervene with a drug that's easier to develop pharmaceutically. And so the drug is called AST-120. It's not approved in the United States, but it's used in several Asian countries in chronic kidney disease. And what's really interesting is 4-ethylphenyl sulfate and similar phenolic molecules are elevated in people with chronic kidney disease because their kidneys are failing. Again, these are urinary metabolites that we excrete, and when the kidneys fail, they build up. And AST-120 is given to these people to help with the buildup of these toxic molecules. And often the consequences of chronic kidney disease could be neurological conditions such as anxiety and brain fog. And so what AST-120 is it's a form of activated charcoal, but it's a proprietary form that is specially engineered to bind this class of molecules. And what's also interesting is that the drug itself is not absorbed. It does not enter into the bodies of mice or of humans. And so as it's passing through the gastrointestinal tract, again, taken orally, it's binding these small molecule toxins, and then as it binds them, and as the drug is excreted, essentially lowers the concentration in the intestines and lowers the concentration in the circulation. And we showed this in mice, and we actually showed that the drug can overcome the behavioral effects of 4-ethylphenyl sulfate. So this is the open field test and the elevated plus maze that I've already introduced to you. And these two first bars are the data that I've already shown you. These are the animals exposed to 4-ethylphenyl. These are the mice that were not exposed to this microbial metabolite. And you can see the mice are more anxious in the open field test as well as the elevated plus maze. But what's quite interesting is that when we give these animals the drug, even though the bacteria in their intestines are making 4-ethylphenyl, the mice overcome their anxiety in both of these tests and overcome many of the other behavioral conditions that these mice have. So again, we're binding the molecule, these toxic molecules with a specific drug. And this study enabled us to go into a clinical cohort to test some of these basic research findings in people. And so I started a company in 2016 around this technology called Axial Therapeutics, and Axial's role is to essentially take the basic discoveries from our laboratory and translate them into people. And so the really talented team at Axial devised a clinical trial in Australia and New Zealand. The demographics are here, 26 individuals, all adolescents. That's what their regulatory board allowed us to do. They wouldn't allow us to go into the younger ages initially. And you can see that most of the individuals who were enrolled in this trial had severe autism. And then compliance was terrific. Over 95% of the individuals took their medication, meaning that over 95% of the doses that were given were actually taken by people. This is the trial design. And so there was a 4-week evaluation where the enrollment criteria was a prior diagnosis of autism as well as gastrointestinal symptoms. But we retested these individuals with, again, standard assessment tools and then took samples to look at their microbiome and their metabolome. If they met the criteria about autism diagnosis with gastrointestinal manifestations, based on our assessment, they were enrolled into the clinical trial where they were on drug for 2 weeks at a low dose, 2 weeks at a medium dose, and then 4 weeks at a high dose. So it's an 8-week trial. And then we took samples from the beginning of the trial. So just remember, visit 2 is baseline or the beginning of the trial, and then visit 5 is the end of the trial. And then we also asked them to come back after 4 weeks and then retested the individuals. And so again, I'm going to show you data from people as they entered the trial and at the end of the trial. And so the safety profile was exquisite. There were really no safety issues, and this is what we expected because, again, the drug had been given to people, but never to this patient population. It had never been tried in individuals with autism. So it met the safety criteria, so that was one of the primary endpoints of the trial. And one of the other primary endpoints was target engagement. And what that means is, did we see a reduction in some molecular or cellular phenotype associated with the pathway that we were studying? And so we met that criteria as well. So target engagement in our study meant lowering these microbial metabolites based on the research that I've shared with you. This is, again, 4-ethylphenyl sulfate. As I mentioned, there are a number of different molecules associated with 4-ethylphenyl sulfate in terms of the structure, and all of these are produced by bacteria. And you can see in all the individuals in the trial, after 8 weeks on drug, that the levels of these metabolites were greatly decreased in their circulation. This is plasma data. This is just a control molecule that does not bind AST-120, does not bind the drug, and you can see it's not decreased. So quite gratifying that we met both of the primary endpoints of the trial. The trial, again, it's a very small trial of 26 individuals, and I should also mention that it was an open label trial, meaning that everyone knew that they were on drugs. So we're not controlling for the placebo effect, which is very, very critical in being able to make claims about efficacy. And so even with this small population, we did see improvements in 2 different behaviors. The first is anxiety, as measured by the Pediatric Anxiety Rating Scale. So this is the entire cohort of 26 individuals at baseline and at end of trial, and you can see there's a decrease in their anxiety scores. But this didn't reach significance. And the reason why is because everyone who entered the trial doesn't have anxiety. About 50% of the autism population is diagnosed with anxiety based on the PARS. So when we take those individuals who had clinically meaningful anxiety, and the PARS, a score of 10 and above means that there's anxiety, you can see that enriched on this population that most of those individuals in 8 weeks reduced their levels of anxiety. And this is the same with irritability. So we use the aberrant behavioral checklist using the irritability subscale to look at the entire population. And here across all individuals, we did see significant decrease in irritability or aggression. In the PARS, a score of 15 is considered, in the ABC, a score of 15 is considered clinically meaningful in terms of irritability. And you can see the majority of the individuals that had irritability going into the trial improved after 8 weeks on drug. And so again, we're quite pleased and excited about these results, but still very early days. We still need to control for that placebo effect that I mentioned, which is a very big confounder, especially in neuropsychiatric conditions. And that's why we've now started a phase 2 trial, which will have both a placebo control and has multiple doses. And what's really gratifying is that just a couple of weeks ago, we were able to get FDA approval to go into younger populations. So now this trial is enrolling individuals at 5 years of age and above. We have 27 sites total, 24 in the US, 3 in Australia and New Zealand. And you can actually find the trial at theautismstudy.com. It's surprising that that domain name was not taken. It's called the Tapestry Study. And again, it's enrolling now. We have actually 2 sites in Los Angeles for those that are interested, but again, sites all across the country. The study will read out in the early phases, probably the first quarter of 2024. And so we'll have then a pretty good idea of whether or not this drug is working in individuals with autism because we have that very, very important placebo control. And I'll just leave you with this final concept that what we've learned over the years with this and other research in the laboratory, again, all based on very collaborative efforts, both here in Caltech and with institutions across the world, is that maybe this research is telling us that one can think about drugging the gut instead of the brain for various different conditions, including neuropsychiatric conditions such as autism. And so the current strategy to address neuropsychiatric, neurodegenerative, and other neurological conditions is to get drugs into the brain, which means that those drugs have to cross the blood-brain barrier. This is very challenging to get enough of the drug into the right parts of the brain. But what it seems that has happened is that bacteria have figured this out. They figured out exactly how to get molecules into the brain from our intestines. But what the research has told us is that maybe we can intervene at the level of the gut. And in those cases, and it's going to be, it's not going to be in all conditions, but in those cases where the intestine and molecules from the intestine have a role in behavioral other neurological outcomes, perhaps we can just deliver drugs to the gut and break this cycle, break this path between the gut and the brain. And obviously getting drugs into the gut is far easier than getting drugs into the brain and much less likely to cause side effects or other deleterious outcomes. So again, we're very, very excited about the concepts, not just the data. And I'll just conclude by thanking the people who did the work. The majority of the work that I just mentioned in the newer studies were performed by Brittany Needham, who was a postdoc in our lab and now has her own laboratory at Indiana University. These are some of the other members of the lab that directly contributed to this research, but our lab is very collaborative. Everyone works together in trying to make some of these breakthroughs. These are the funding sources that helped support this work, and these are the many, many collaborators, again, both here in Caltech and elsewhere, that really enabled us to do these cutting-edge research. And of course, a lot of thanks to the families and the individuals with autism, both in the first phase trial as well as those that are entering the trial now. Thank you so much, and I'm happy to take questions.
Thank you so much. Thank you so much. Because we went a little long with the intro, we're going to do 5 minutes of questions. This will not end right at 9:45. So if you need to use the restroom, you can. We'll start the next session at 10, but we're going to do some questions. And thank you for these great questions. Edward and Kutulenk ask, do you feel, and there's a couple of questions that are ganged around the environmental issues, do you feel that the rise of microplastics being introduced into our food chain, this is our digestive system, could be a contributing factor to the rise of autism? And there's a lot of questions about if you recommend people taking probiotic products.
Sure. Whether it's microplastics or any other environmental exposure, we simply don't know, right? And there's just simply not enough data to understand what it is about the environment that may be changing the incidence or the diagnosis rates of autism. And again, I want to be clear that the role of the environment in autism still remains unproven and controversial. It's research like this, and I want to say there's many other researchers around the world who are identifying both microbial and non-microbial, meaning environmental, contributors to autism, largely in animal models, but some translation now in people. So again, whether it's pollutants or other toxins, food, preservatives, antibiotics, and certainly not vaccinations, but other chemicals that we're exposed to, the research just simply isn't there yet to understand what are the environmental contributors to autism.
Pete K. and many others were interested in what is the mean age and age range of autism diagnosis in the US, and do you have a sense of how this has evolved over the past couple of decades?
The mean diagnosis age is about 2.5 now in the US, and that has decreased over the years. It was 4 or 5 years of age about a decade ago, but again, it's not attributed to any changes in the diagnostic rates, but just the fact that physicians are just more aware of autism than they were a decade ago and prior to that. And physicians are just looking out for it, right? Because in many cases, it could be quite subtle. In some cases, it could be very obvious. And so the decrease in the mean rate of diagnosis is just because society is more aware of this condition.
And Jim Turman and others are interested, should we expect a spike in autism cases from children who gestated during COVID-19?
Yeah, no, it's a great question, and those studies are underway. Obviously, society provided a great experimental setting, a clinical trial, if you will, in society that many people are now following. And so they're following those pregnancies during COVID, those individuals who may have gotten COVID during pregnancy, how severe their inflammation and fevers were, and then now tracking the offspring to see what are the rates of diagnosis. It's going to take several years, certainly, to get that data, but many groups around the world are tracking this.
Our very clever Caltech community is interested in, of course, the matter of, I'm going to gang these questions, with an increase in monitoring genetic content of human waste streams, so it's possible to look for B. fragilis in the human waste stream across time and geography, and can 4EPS be detected in wastewater?
I don't know. Certainly we can detect Bacterius fragilis, we can detect any organism in wastewater. But again, the molecule, I don't know. But again, the organism nor the molecule is new to humans. It's likely been with us for many millions of years. Why high levels have evolutionarily resulted in these changes in behavior, I think maybe that's a different question. And that's not certain. I mean, is this an evolutionary accident, or did somehow bacteria intend to cause these effects, and what so evolutionary forces that drove these interactions were? Again, I'm afraid to say are just not known.
What? You're at Caltech, you should know everything. All right, Gary Turner and some others are interested, if only about 1/3 of people with autism have excess 4EPS, why were the clinical results so good?
Because the molecule itself sequesters, the drug itself sequesters many different molecules. And so there are likely other molecules that are binding AST120, binding the drug, that are similar to 4EPS, but may have different properties, and those just haven't been studied yet. And so there's, to the best of our knowledge, about 13 to 15 of these molecules that are related to 4EPS that the drug binds. And so it's likely that some people, again, assuming that the model and the hypothesis is correct, that some people's symptoms may be driven by 4EPS, other people's symptoms may be driven by other molecules, but the drug binds them all.
And last question, I think ganging together a couple of other medical conditions, any potential relation to Parkinson's treatments or any connections of gut to Alzheimer's?
We've started now looking for these molecules in Parkinson's patients and in Alzheimer's, and they do seem to appear. The effect isn't as high as they are in neuropsychiatric conditions. So I can tell you that these metabolites appear to be elevated in schizophrenia and PTSD. And so there may be differences in what types of neurological conditions that these molecules are associated with. But if I were to venture a guess, I would say that the pathways that, by which the microbiome impacts neurodegenerative conditions are likely going to be different than the pathways by which it's influencing neuropsychiatric conditions.
Thank you very much, Dr. Sarkis-Mazmanian, and fantastic questions, fantastic presentation. We'll take a quick break, start the next session right at 10, Our Quantum Future. See you in a moment. Thank you.